Hi. Our smart scans are tailored to each customer and their specific strategic objectives. Here's an example of a smart scan for offshore wind power. This example includes a summary of key developments, key market developments, the latest innovations and technologies, and other information of relevance to the specific use case. We can structure and deliver the information in many ways so that it fits your way of working, and once configured, the smart scan is automatically updated with the latest information. Our platform can also use the monitored information to generate a range of new content such as podcasts or a summary report on a topic. Imagine you ask for a report on energy technologies. Our AI assistant will then guide you to ensure the report meets your requirements, and within seconds, a report is generated for you, covering the latest information we have collected on the topic. This way you can interact with and extract highly relevant insights tailored to your business. Get in touch with us to know more about our different solutions. Thanks for watching.
My name is Lars Tvede. Today we are going to talk about biotech, and especially how you use knowledge about biotech to do production. And I have a guest, which is Stefano Bertacchi, I have to say that right, who has been a youth ambassador in biotech for the EU, which is not something everybody can be at all, and who also has a PhD in biotechnology production. And for me, this is the whole subject is super important, but it's also super fascinating. Just a few weeks ago, I finished writing a book about the history of intelligence, history and future of intelligence, going back from the Big Bang. And so one of the things we came across was the complexity that biology has created, and we went into how many different eukaryotic species exist now, and how many have probably existed throughout the history of life. And then we made the best estimate we could at how many different base pairs have there been in all the different DNA. And our estimate came out to that if you make a bookshelf, which is 2 meters tall, and it has 8 levels of books, and every book like book, a book like this is simply filled with genetic code. And we took all the genetic code that life has produced so far in the history of Earth. That bookshelf would be, I have to ask you, Stefano, because you don't you don't know the answer. How long would that bookshelf be? Bookshelf. Just repeat. A bookshelf. It's 8 meters tall. There are 8 levels of books back to back. It's only the genetic material that has been produced by eukaryotic species throughout the history of life.
So going up.
No, wide. It's 2 meters tall. That's really,
like 2 billion kilometers.
20,000 kilometers, like halfway around the world.
Okay.
Yeah, so that's the genetic material that these species have been produced. But then you have bacteria, and nobody has any idea.
You were thinking just eukaryotes.
Yeah.
Sorry,
Yeah.
I overestimated because of thinking of
Yeah,
bacteria and
bacteria.
viruses as well.
Yeah. So nobody has any idea, as far as I can see, how many bacteria exist today. There seem to be bacteria 40 kilometers up in the air, which is where there's pretty much no air. And also, probably 8 kilometers, something like that, into the ground where it's super hot.
Yeah.
There are special bacteria that grows inside the Chernobyl reactor. They actually like radioactivity. And there's a growing theory that there are actually lots of very simple extremophilia microorganisms around in space, floating around in space, looking for a place where they can start to grow.
With this, I published a book on the topic of microbiology and microbes, and it was called Little Geniuses in Italian. That's because I like it to say that microbes are really geniuses in the sense that they can colonize really a lot of parts of our world, and they will be the first life forms, and they really are uh, tremendous, tremendously able to survive and to thrive, and we can learn a lot from there studying them.
Yeah, and probably, I mean, once you have life on a planet, it's almost impossible to get rid of it because the microorganism can adapt in unbelievably. But then, so now we're talking about the genomes of them. So, fun fact about that, of course, is that my dad was a chemistry engineer, and I'm just thinking about when he took his education at the university in Denmark. They had no idea. Nobody had any idea what actually was driving life. And so
Yes.
that was my dad. And the first very grainy picture of the structure of the DNA came in the 1950s, didn't it?
Yeah, exactly.
Yeah. How did they do that? How did they take that picture?
Yeah, Rosalind Franklin was able to take that picture with X-ray crystallography with
Right.
a very
Crystallography.
complicated process that was used to obtain the 3D structure of the molecules,
Yeah. Right.
even things. Now, luckily, we have, like, for example, tools like artificial intelligence to have much more of that information. But that work from Rosalind Franklin was terrific because she managed to provide the structural information on the DNA at the same time,
Mhm.
understanding that was understood uh, briefly before that DNA was the genetic material, the one
Right.
that provides the information to how to make a cell. But we are talking about the 50s. So it's something that is not even 1 century old. Now we have
Yeah.
these tools to play with the DNA easily.
So they and there were there's actually there's a super good book about it because there were two teams racing right to find it first, but those two did it. But then, I mean, that information about that structure is less than a trillionth of what we know
Yes,
for sure. Yeah,
definitely.
today, and and then. Oh, and we're not even talking about viruses, which is not DNA but RNA. So that's like,
No, there's viruses made of DNA as well.
okay, okay,
They are both. There are some made of DNA and
right,
some made of RNA.
right. Um, Probably RNA came before DNA actually, because RNA is simpler than
That's debated. I mean, the
yeah.
concept of the RNA word that says that RNA was like it created itself before DNA. It's debated again. We are not really sure about that again because it's complicated to go so back in time to understand what happened. What we know that of course DNA was one of the first, and there will be like around other molecules, but
Mhm.
thinking about that, I mean, how cells work, it's definitely incredible to think that evolution provided us to the things that we see today, and how many like trial and error was made during evolution, and things that we lost just because they were not the fittest one in that specific moment. But luckily, with biotechnology, we can now explore all the things, considering the things that we need at the moment.
Yeah. So my understanding of the the most widespread hypothesis of what happened is that you had these geothermal vents at the bottom of the sea, which is where you have super hot water coming out from the sea, and then it creates this cone which can be up to 30 meters tall. And so you have colder water outside, although it wasn't very cold. It was in the beginning probably 50 degrees or so.
Yeah,
But
kind
there's
of.
super hot, superheated water inside. And then you had clay, and with lots of very porous clay. And then inside that, you would get micelles, which are underwater bubbles. So you think of a soap bubble that is also has water inside and outside. And then you have
Minerals,
uh,
definitely.
yeah, you had minerals, and you also had because of the temperature difference, there was also a acidity difference, which turned the whole thing into a low power battery.
Hmm.
and so you had kind of electricity flowing through. So you have a power supply, and then inside that you had these precursors of RNA that could create each other and copy themselves. And so some of them would get encapsulated into micelles randomly, probably all the time. And then eventually, and that was very, 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 I mean, that took a long, long time. But then
No,
some of these
actually,
became,
we can imagine cells as just a bag of proteins, enzymes, and nucleic acid in forms of DNA and RNA that are just happen to be able to
yeah.
replicate its own information, and that information is a code that can be translated into proteins. So definitely, it's something really really interesting, and the idea is that like completely by chance a bunch of these molecules happen to
Ribozymes are
find
the key,
themselves
right?
into the same environment, same closed little environment. So I
Yeah.
always like to say to my student that basically the microbe cell is just a bag of enzyme. We can think about it that way. Unfortunately, a lot of enzymes, and in fact, sometimes when we have to handle with chemistry people, that of course are one of the biggest like partners in developing processes, it's hard to make understand that it's not just like one, two, three, four reactions that in chemistry we are more accustomed to, rather than tons of reactions that occur at the same time, and you would
Mm.
like your cells to do just the thing that you would like to, but they are doing a lot of things that for you in that moment are useless. So also going towards the minimal cell is something that is happening, like to have like the minimal essential luggage, to have an empty shell that is just able
Mm.
to replicate itself and thrive, and then you just add the things that you need without like the noise of the other reaction that can like make you consume the raw material and waste a lot of money also as well.
I have something. We have to get to the production, but I have a funny thought. So I would think that the minimal viable cell has somewhere between 120 and 150 genomes. I mean, people, you can discuss that. So we know that the complexity of DNA, which is complexity is a little bit of a fluffy word, but it over time it doubles every 320 million years, I think. So if you run it back from what we have now, the most complex species like ourselves, we need something like 9.6 billion years to go back to the minimal viable cell. And the problem with that is the Earth is only 4.6 billion years old, which could suggest that life on Earth actually was imported to Earth from somewhere else. So it's cells that were created on some other planets and were, dispersed into the universe and then randomly landed on our planet. But that's a bit of a side story. So where we are now is that it's so complicated. I mean, biotechnology is so unbelievably complicated. But then we got AI, and just within the few years, that has completely changed some of the situation. Because if you take a protein, so almost everything, almost every complication in life is about proteins. I mean, apart from the DNA itself, and proteins. One thing is that you have a description of the chemical composition of a protein, but what a protein can do depends entirely on how it folds. And so it folds like a spaghetti, like an Italian spaghetti. Some of these they're absolutely huge, and the only part of the protein that really at any given time can interact with the surrounding world is the one on the surface. So we have all these different structures where it can fit to another one, react with another one, and so on. And it used to be until very few years ago that, for instance, a PhD student could spend a whole year or 2 years, a whole PhD project on figuring out how one single protein would fold. And then comes DeepMind from Google, and then they put AI to the task, and then they can do this 5.3 million times faster, something like that. So there are different programs doing this. One of them estimated the folding structure of 600 million proteins in 2 weeks. And now you also have software that can estimate what happens if you take one protein of one spaghetti and put it next to another one. How can they interact? Of course, that's super important in figuring out um, what happens in the human body if we introduce this, or we have this problem and we add this medication, for instance. And the number of permutations are absolutely enormous, but AI is chewing through this. And then the other thing it can do now is that it can say, we need a solution. Let's say there's a specific kind of cancer that has a specific biomarker on the surface of a cell, and we need something that can attach to that and mess up the cancer cell. And then we have never found a protein that can do it, but software can figure out how that protein should look, and then it describes that protein. But then you have to pick up your phone and call Stefano, and then say, Stefano Bertacchi, I need a protein. Here's a recipe. Cook the spaghetti for me. Is that? And then you have to go into bioproduction and figure out, how can we make a microorganism that can create the protein that I need to cure that cancer? Am I is that right what I'm saying?
Yeah, yeah, of course I can cook spaghetti very well because I'm Italian as a start. That's the starting point. And then, yes, I mean, I'm working with the development of bio-based process, so biofermentation as well, using microbes, also genetically modified ones, to obtain compounds, molecules, or materials that are of interest. So among those would be like an enzyme. At the stage, I mean, the thing is that I might say that the hardest part is to have the sequence, the DNA sequence of the enzyme, of the molecule, of the protein. Let's say that your product is a protein. Let's make the easiest
Yeah,
option.
I want a specific protein.
Okay. The thing is that
I've given you yeah.
okay, as soon as
I,
you
so
have
I've given you a specification of protein that I know that protein folds in a way that it has a chemical structure that I can use, and it has a way of folding that I can use. So I
okay.
tell you, can you make that?
Okay. So what I would do is
What
like
would you do?
having the reverse. So you have amino acid composition in the order. So I make it a gene. I make a gene specific for the microbial host that I would use. That it could be a bacterium or it could be a yeast, for example. You have some good yeast that are
Right.
able to expel proteins for protein production, like monoclonal antibodies, let's say. So I would like buy a synthetic gene. So there are these companies where you just send them the sequence that you want, and they produce it, and they send it in a vial. It's kind of cheap also. Now it costs just some euro cents per base pair, becoming very,
Right.
very cheap. And so with few hundred euros, you can have the sequence of the protein that you would like to have. And then you insert it into a circular DNA to insert into the bacterium or the yeast.
Yeah. Okay.
Um,
So can I just stop you here? Okay. So you get from this laboratory, they send you, what is that little bottle that contains the genes?
yes, it's
Yeah.
just a little vial, a little
Plastic bottle.
bottle,
Yeah. Yeah.
let's say, which has like lyophilized DNA, which
Yeah.
is the DNA that I ordered,
Yeah,
basically.
you ordered that DNA, and then you
Yeah.
have a culture in some container of yeast, for instance, and of
Yes.
course inside every yeast cell inside it has DNA.
Yes.
You wanna insert from that bottle
Yes.
those genes into the DNA of those living cells.
Yes.
How do you do that?
Yeah, so different methods possible. One is involves the use of some compounds that create some holes in the membrane. You have to imagine a cell with like walls outside,
Yeah.
that is the membrane, and you have to tell them, okay, listen, make it enter.
Right.
So to oblige them, you can use the force, and the force is like using some compounds that create some holes, or at least weakens the membrane to make the DNA from the outside enter. Or if you want also to be even meaner, let's say, you can use electric shock. So you can put like in a specific, let's say, bottle, and you
Mm.
charge with electricity, and this electricity creates some holes in the membrane, makes it easier to enter the DNA. And
Does it?
then it's,
Excuse me.
yes,
I just have a side remark. Have you ever heard of a gene gun?
yes, it's the
I
gene
was
gun,
thinking.
but the gene gun is used mostly for bigger things like plants or animals.
Yeah. So
Because
I
if you try to shoot to a yeast, it's complicated too.
right. So I
Actually,
just want, first time I heard about a gene gun, I sort of laughed. So I just want to say, so it's actually a gun that shoots genetic material. So you used it a lot in the beginning in genetic engineering, as I understand, on plants, that you simply shoot genetic materials on the plant and then hope some of it would randomly penetrate cell
yeah,
walls.
actually, it's a kind of. I've never used it. From what I remember, it's like on the ballet.
Right.
It's like covered with the DNA you want to insert,
Right.
and the idea is like that while shooting to a leaf, for example,
Mm.
by passing through it will some enter
Some of
the
it. Mm.
the cells. But from what I remember, it's not very efficient. For example,
No.
with the plants that are other method like using a bacterium which is naturally able to genetically modify a plant. So
Right.
by
So the fantasies I had that people I don't like, I could have genes from a fly, and then when I pass them on the street, I shoot them with a gene gun.
and they fly away.
No, but anyway, so let's get serious here. So, okay, so you get the genes inside the cell. Does it need to integrate with a genome, or can it be a plasmid
Okay,
that flows freely?
it's two options. It's either the two are possible. So it's a plasmid that keeps being outside of the genome, and then other options is that you integrate into the genome of the host cell. I mean, there are pro and cons of the two. It depends on the host. It depends on how you want to induce the production of the protein. I mean, when I did it with yeast cells, I integrated in the genome. When I did it with bacterium, I left it into the plasmid, let's say. So I
Okay,
produce a protein with both. So it really depends on the host, let's say.
so I have a question again here. So, the Moderna COVID vaccine was RNA, right?
Yes.
And that was in the form of plasmid, so it was free floating, and then it breaks down after a while. Is the difference that when you integrate it into a genome, into the existing genome, then it becomes stable, but if it's free floating, then it breaks down after a while, or
Okay.
is it difference because you're talking
So
about getting it
two
inside
things.
the cells? Yeah,
Okay,
go
two
ahead.
specifications. So first of all, the vaccine, the RNA in the vaccine is not plasmid. It's not
Ah,
circular. It's RNA.
okay, okay, all right, okay,
And then the problem of the degradation of RNA is the fact that RNA is weaker than DNA.
yeah, because
So the thing is that
doesn't have a
the
self
thing is that
copy.
exactly. So also because it can be attacked from the RNases. But the thing is that the plasmid that we use to insert into the cells, it's made specifically to stay
Ah,
because it
okay,
has a replication origin, so to be replicated. But then the issue is the selection. So you have to like convince the cell to keep it, and sometimes you use like a marker, for
mm,
example, resistance to an antibiotic. So I put that gene. So in the plasmid, there's the gene that you want for the protein that you
mm,
want, and for example, the resistance to ampicillin, which is an antibiotic. So this means that in the presence of the antibiotic, you have to keep it. The plasmid, otherwise you die. So the way
mm,
to select the cells that kept having the plasmid is to have a selection from the outside. that you don't have when you insert into the genome. So one of the pro of inserting into the genome is that it's there, so it cannot go out. But at the
Yeah.
same time, one con is that you have one copy. Otherwise, when you have a plasmid, you can have multiple copies. So you can have multiple genes and produce, in principle, more proteins.
Right. Okay. And RNA is what creates proteins. So you're now created and inserted into a cell something that creates the protein that I want from you.
Yes.
How does that protein get out of the cell, though?
Okay, so there's two ways. One way is that it doesn't get out of the cell and you have to break the cells.
Right.
But that could be problematic because sometimes they tend to cloth together because there are
Mhm.
many. At the same time, also there's another issue that yes, you want this cell to produce the protein, but you don't want to make it feel bad because otherwise it doesn't produce it. So you can produce it a lot, but not too much because
Right.
then it doesn't grow. Okay.
Mhm.
So you can either break it, so you centrifuge the cells at the end and you break it. You have to isolate it, and it could be in a way that I'll explain you later because it's the same like the other one. Otherwise, you can have like specific in the plasmid, for example, that I was telling before. There are specific already signal sequences that tells the cell to export outside of the cells.
So
So
it's sweating the protein I want.
exactly. So
Right.
basically, you insert into this plasmid that is already made,
Mhm.
where you have like this additional part, generally at the beginning, and the protein is produced with this part, and this part tells the cells, okay, bring me out. And so it's transported outside, it's then carried out and transported outside of the cell. And then in that way, you have your protein outside of the cell. So in principle, it's easier because you don't have all the things that are inside of the cells to get rid of. But anyhow, you will have to purify it anyway. So there should be like a specific way to purify it. And one of the most common ways to have like tags. So the
Like what?
proteins
Like
tag,
what?
a tag.
Attack.
So you have like at the end of the protein a specific sequence generally made of histidines, that is an amino acid, that you add in the synthetic gene,
Right.
and then you have like specific. You take like the liquid, or the liquid of the cells that were growing on, or the inside of the cell, and then you pass through this like column that is a kind of a filter, let's say,
Hmm.
that
Yeah.
keeps it's specifically made to retain all the proteins, only the proteins, and have this tag, and
Right.
going through all the others. Okay, and then you cleaned it up and you have some fractions where you just have, or mainly you have your protein concentrated.
Okay. So now you have protein. You have concentrated the protein, and then I basically shoot it into a patient that have that cancer, and then that protein can attacks that cancer. And so I can imagine that something like that is very expensive. It can cost, I don't know, 20,000, 100,000, $300,000 to cure that, or try to cure that patient. But these things are getting cheaper in mass production. So what I understand is, for instance, I come from I actually I worked in the dairy industry when I was young on Iceland. We made cheese, and I was the first one showing up in the factory in the morning. I would fill up 5,000 liter of fresh sterilized milk in a tank, and then we put half a liter of bacteria, or I put, and then I put a little bit of rennet, which came from cow stomachs. And then within a surprisingly short time, the whole thing was acid and or began to get acid, and then it became a solid mass you could cut. And this rennet was expensive because you would extract it from cow stomachs. Of course, soon after they started to make it artificially with precision fermentation. And rennet was as because it was expensive to make, that it was great business to do it in the other way. But then it's going to become cheaper and cheaper. You can make yeah, you have to tell me, but you can make um, elements of cosmetics. You can make elements of food. you're beginning to make materials, and we are perhaps on a threshold where we begin to make food itself. I mean, the bulky food through precision fermentation processes. I mean, there are already companies working on it. Can't you take us through some of the things that have been super interesting and some of the things you think will become super interesting in the foreseeable future that you can produce with these technologies?
So definitely, RenNet, it's one of the products that we can now make with microbial fermentation. That definitely is useful when talking about, for example, vegan cheese, because RenNet originally comes from the animal,
Right.
from calf. So therefore, if you really want 100% of the pipeline of being animal free, it's something, and often derived from genetically modified microbes. So again, we see that all those topics related to sustainability or having a healthier food and so on, it's related to GMOs, despite the fact that people is still afraid about this topic, especially in Europe. Um, Definitely, also other compound, other drugs, let's say, that we have easily access to. It can be, for example, the enzyme for the degradation of lactose for people lactose intolerant. That, of course, are many in the world. Actually, there are more people lactose intolerant rather than those that can digest it. That's one,
Mm.
because in Asia there's tons of. Definitely something that is really exciting in the world of precision fermentation, and now we call the technology to use microbe to produce specific compounds. And actually, what I was already doing with industrial biotechnology, it's to obtain proteins that can alternative proteins that can be used for the food sector. So to detach from use of animal proteins. There are already microbes that, as they are a source of proteins like spirulina bacterium, that actually is a bacterium, not an algae, but is
Mm.
advertised as an algae. Or other molds or yeast that are used in other cultures, especially in East Asia, like those for the production of tempeh, or fermented beverages that are definitely interesting for different reasons. But then, like for example, there are some companies that are interested in recreate the taste of meat, because one of the like complaints of the consumer is that vegan meat doesn't taste like meat. That regardless of
Mm.
the fact that it doesn't should have, but it's not the
Mm.
story. The production of heme, which is the protein, I mean the part of the protein that provides the with the hemoglobin, so the taste of blood, let's say, that
Mm.
gives a distinct taste to meat, has been now produced with recombinant yeast, so can be used specifically to as an addition to food to make it taste like real meat. So there's a brewing, sorry the joke, interest on the growing of microbes for a lot of types of compounds, like for example bioplastics. I've been working with the production of bioplastic compostable and biodegradable bioplastic from an engineered yeast, for example, or the production of carotenoids, which are the molecules that provides the orange color to carrots, but are really interesting for different sectors, from the food, from the feed sectors, and the pharmaceutical and the cosmetic ones as well. And generally, they are produced from petrochemical synthesis, so with all the problems that arise from that. So having the ability to have a microbe that is naturally able or engineered able to produce this kind of compounds, definitely we can provide us the ability to detach from the use of fossil resources, and to boost bioeconomy, because then again, as mentioned, I've been appointed as EU Bioeconomy Youth Ambassador. So also the push for more bioeconomy and the valorization rather than blind exploitation of biomasses and renewable bioresources is definitely something that is necessary to cope with and to survive to all the crises that we're living on. Definitely what I see in developing is also the application of synthetic biology tools on in these fields. We have seen that we were discussing about the DNA just being discovered in the 50s, but technologies like CRISPR-Cas have been developed in the tens of the
Yeah, this
2000.
which is for editing
Exactly,
DNA.
gene editing, and now I'm using currently. I'm not saying every day in the lab or something like that, but when I studied at university, it was not so many years ago. We didn't even study it, and now I'm doing it from several years, both for gene editing, but not only also for transgenesis. And this really revolutionized the way that we are doing genetic modification because it's similar to what we were doing before, but faster, and we do more things easier and faster with a more precision. And for example, in the agricultural sector, it's really revolutionizing because we can go beyond the common transgenesis with gene editing of plants for doing a lot of things. And then it goes then on the topic of the regulations that are the boring stuff, but of course they are relevant for what we can do or not doing. For example, in the EU, the regulation of the new genomic techniques where gene editing is involved is really important because it would decide whether they are to be considered GMOs or not, and then it has downhill implication both on the communication of this topic and the acceptance of the from the audience. That's why I'm also active from the scientific communication point of view on social media, on events or webinars or books to explain people this technology that, as you mentioned at the beginning, are really complicated and really technical. So again, when I deal with this topic, I don't even know with the general audience, I don't even know if I can give it for granted what genome is or not. So of course,
Mm.
like starting from scratch is always complicated. And also dealing with companies, I often deal with professionals of other words, especially the chemistry one. It's hard to make them understand why this technology related to fermentation and biotechnologies in bio are really relevant and can really transform a lot of enterprises. Unfortunately, in Italy, I may say it's more complicated because we are less prone on like groundbreaking innovation and changing the things that has been like that for many years. But I see that other countries are really pushing for that. So I might say that at one point Italy has to cope with that as well because of the market.
Italy outlawed cultured meat recently, didn't it?
Yes, actually, they tried, despite being 100% not possible because it's like banning the free traveling of goods in the EU, and you cannot do
Mm.
that because if something is. It was mainly a political move to say that we are defending made in Italy. But I like always like to say that it was like banning flying cars, so something that doesn't exist. So actually it doesn't exist really. So in the EU no one
And, there's a little bit of cultured meat around.
yes, definitely in the US or
Yeah,
in Singapore there were being
mm. Chicken.
exactly
Yeah.
applied for and accepted. But actually in the EU there are some little companies that are doing R&D with that, but it's not something that is ready for the market really. And also, no one. Maybe now there's one from the Netherlands ever applied for the EFSA for approval. So
Mhm.
again, it's not me, it's not you, it's not the politician that will tell
Right.
that cultivated meat is safe or not. We have that agency, that the Food and Safety Agency of the EU is the one has to say yes or no, and then when it's yes, then it can be commercialized in the EU, and not single country can say okay, that's no. Was more a political move that I mean I would say it was a perfect political movie because it was just you were banning something that doesn't exist.
Right.
So
And
you
then
are not losing anything at the moment.
okay, can this, we have to take questions in about 5 minutes, I think, but I have a couple of questions. So if we dream, uh, so you have everywhere where you have farming, you have problems with fertilizers, so you fertilize the plants, all good. Problem is some of this fertilizer goes into the water and then it goes into the rivers and the lakes, and then it stimulates growth of too many algae, and then you have lack of oxygen there and so on. And you do it mainly, I mean, you do it partly for phosphorus, but you do it largely for nitrogen. Because when you keep removing the plants from the land instead of they just rot and it kind of circulates, you remove nitrogen and then you have to add it. And the very old days, the nitrogen would come from poop, and then they didn't have enough of that, and then they found guano on some islands in the Pacific. Then they didn't have enough of that, and then smart German, I think it was invented. This is the Haberlab process, which extracts it from the air, and about 1% of the global energy consumption is actually used for that. But if you can genetically engineer the most common plants so that they do that process, which some plants do it, then we wouldn't have to fertilize with the way we do now, and we would solve a huge environmental problem. Is that doable? There's a political or legal resistance to it, or can we do it for? I mean, there's not very many plants. It's wheat, it's maize, and so on, rice that feeds the world.
Okay, so the Fritz Haber process for the production of nitrogen, ammonia from nitrogen in the air, as I mentioned, is really, really uh, energy intensive. So there's a lot of ideas to how to cope with that problem. So there are many options. So one option, as you mentioned, is the one genetically modify the plants. Then it depends, because if it's a GMO, then it goes into the regulation of GMOs, and it's
Mm.
politically possible. It depends where you are in the world. If it's the US, there's no problem. If you are in the EU, in principle, it's not a problem, because then EU is not banning the GMOs, because I don't think people think that EU is banning GMOs, but it's not true. Basically, they accept or not, and then the single countries can decide whether to accept them or not. Then the problem is that most of the countries do not accept. Italy first row, but like Spain is producing GM corn. Okay,
Mm.
so is it possible? Yes. Then we have like the gene editing. Again, then it depends if it's considered GMOs or not. That is in the European Union, we are having a debate on that, and now what is suggested by the Commission is that they should not be considered GMOs if they are modified in a specific way and you are not inserting an external gene, so it's not transgenesis. But then other options that you might have is go back to microbes, because actually the plants that can do it thanks to microbes, like legumes, like beans that are really rich in proteins. Are this way because they have in their roots microbes that like have a cheat code that permits them to catch the nitrogen that is in the air, transform it into ammonia, and pass it to the plant. So in principle, something that can be done, it's microbial fertilizer to enhance the presence of microbes that are naturally able to do that, or modifying those microbes to make it more ammonia available to the plant. So it's either touching the plant directly, or either touching the microbe that live in the roots of the plant.
Okay, the other question, please just give me a super short answer because we have to take the questions. Uh, but it is obviously super attractive if we can make plastic that is biodegradable in different ways that we determine through precision fermentation. And my question is simply, when you do this precision fermentation, for instance, for plastic, we're talking about potentially millions of tons produced every month or something like that. What do these microorganisms eat? The yeast or the bacteria? What do they eat? I mean, it doesn't.
To produce the plastic, you
Yeah,
mean?
to produce anything really, to produce food or plastic or proteins to cure cancer or
The,
anything.
the,
What do
the
what?
answer is easy. It's sugars, generally.
Okay,
Sugars
well,
or proteins.
okay, not healthy for human. Works for.
Better from residual resources. So generally, we provide sugars from sugarcane or corn. Of course, this is not sustainable because it's from edible crops. So what I do, for example, I give them junk. As I give them junk, like woody biomasses that has sugars inside. The only thing is that I need to crack them to release the sugars, and I use other enzymes that are good in like eating the macromolecular structure and release sugars that are good for the fermentation by the yeast.
Okay, well, we make a lot of sugar and then we put it into these bioreactors and then they can make everything else.
I mean, we are talking about fermentation, in fact.
Yeah. Shall we look at if you can you click on Q&A on your
Yes,
screen? I've done the same, and then there's a question for you
at
from
the
Alina.
bottom.
It's yeah. Can you read it if you
Yeah.
could?
Question for Stefano. You have quite an impressive following base on Instagram. How are you making people interested in biotech stuff? So yes, as mentioned, I'm science communicator. I have 35,000 followers on Instagram. As Lars discovered today, it's mainly in Italian,
Italian.
unfortunately. But my audience is mostly Italian, and the English is not spread, unfortunately. But I mean, I'm targeted on the topic. I mean, we are 60 million, so we are a big audience.
Yeah.
Um, and I try to make people being interested in biotechnology, explaining like the interesting application on the cool things that are behind the biotechnology. And I try to use different tools like memes or funny things, funny stories to explain these topics. And I have a good following base also that for the events in person or the books or everything in Italian. And definitely, I mean, I see that people becomes interested. Some high school students and tell me that they enrolled to biotechnology just because they saw they get interested and involved in this matter following me. Some people also started a PhD because of me. That's hard, let's say, but it's definitely interesting, and you also feel the responsibility because during the pandemics and lockdown and development of vaccine, despite I'm not a doctor and so on, a lot of people were coming to me asking for information about that because it was a mess around there, and I also didn't know a lot of things. So also being humbling to say I don't know stuff and go to the proper references is important.
Yeah, but I would like to add that okay, so Europe is falling way behind on AI, for instance. So there are many areas where China and especially the US, well, both are getting way ahead of Europe. But in pharma and biotech, I think Europe is way up there. And for instance,
Yes.
the most valuable company in Europe, I don't know right now, but at least it's probably still Novo Nordic, which is a biotech
Yeah.
company in Denmark. I live in Switzerland, which is
Yeah.
also
Novartis, of course.
and La Roche. So we have another question. How do you see the role of synthetic biology evolving in the next decade? That's a big question.
Yeah,
You take that.
I see it definitely growing and growing, merging with artificial intelligence technology. Definitely to especially to search for new genes out there. We don't know all the microbes that are in the world. There are many that we have never discovered. But with these technologies, we can really have superpowers in understanding their nature and their genome, and to probably extract new genes that can be easier and faster deployed into microbial cell factories like microbes. And especially with automatic tools for screening, because one of the problems with the beginning of synthetic biology was the fact that you needed to create a lot of combination to find the best one, because uh, at the beginning I said, okay, you put the protein inside the cells and then it's done, but it's of course more complicated than that. You might have combination of promoter, which is basically the switch off on of the gene. So definitely synthetic biology can really have boost with automation of like literally machines that can provide you the screening for hundreds or thousands of different types of combination to understand which is the best one for that specific situation.
So,
Um,
let me just ask you. So, you have kind of two worlds competing here, or working jointly. One is that you scan the nature to find genes, and then when you find genes, you can analyze what they do, or you can observe what they do, and then you can use that for some purpose. And the other one is now that we have AI with vivid imaginations, generative AI, that imagine proteins that could be useful for some purpose.
yeah, also,
Um,
also
and
that
then
one
you
that.
kind of reverse engineer, or you engineer.
Yeah, both. I mean, it's both thing. I mean, you can also knowing like feed the intelligence with the knowledge that we have that like to came up with
Mhm,
a gene that doesn't exist and a protein that doesn't exist to
mhm,
do new things, or to scan what nature has already been done, because we have to remember that nature had like really million of years to
mhm.
I have the opportunity to explore different options.
Yeah, tried a
So
lot.
exactly, tried and failed a lot.
Yeah.
So again, looking at what nature does and exploiting and exploring biodiversity is fundamental. The research that I do, it's funded by the EU funded project that is the National Biodiversity Future
Mhm.
Center, which has the main goal to understand, to valorize, especially Italian biodiversity, but in general biodiversity, and of course microbial biodiversity is one of those. Look for, I don't know, let's say microbes that are able to scoop up specific heavy metals.
Yeah.
So you
Question.
go for places where you have those heavy metals, and then you can apply those proteins to places where there are heavy metals because we polluted it.
Yeah, that's smart and it's very elegant. So, question: have you ever seen the TV series called Pimp My Ride?
Yes, I
Okay,
mean I'm old enough.
so they take a wreck and then they make it into a super cool car. So there is an industry, or there's a discipline which is called gene tuning, isn't there? So you take a gene that is useful, but you want to make it 100 times more active, and then you can tune it so it becomes 100 times more active,
Yes, so
or,
yes, in that way
or
it could
tweak
be
it a bit so
different.
it just does the thing somehow better for your purpose.
Yeah, I mean, the gene tuning could be in different ways. So from one side, to tuning the expression, so as again touching the turn on to the switch um, of the gene expression, or again like touching the protein itself. So um, I mean, just modifying a single brick of the protein can increase the speed of the reaction or can change the substrate. So again,
Right,
another of the challenge is to change the substrate from the original one to another one, because often
right,
the proteins are already promiscuous. Sometimes you want to enhance that promiscuity to make it using one compounds and not the other. Also, there's one of my contents in on Instagram in Italian where I say that discrimination is a good thing, but of course
hmm,
in the sense that proteins often are able to discriminate between two molecules despite being very very similar among each other,
yeah,
which
mhm,
is really important because we can have like a specific target and then it's not touching the other things that are around.
right. You can also, so there's this on Netflix since New Year. You have a documentary about Brian Johnson, who is spending $2 million a year on longevity, and he goes to Honduras and then he gets injections so his muscles grow faster. So that's that's uh,
Okay.
he's tuning himself there. Hope it goes well. There's another question. What emerging trends or technologies in biotech are you most excited about right now? I think that's a really good question.
um, let's say that probably I don't have one specific, probably all new technologies, or I mean, like synthetic tools, really like specific ones, like new Cas proteins that can be used for the genetic editing of. Um, of the genome. So let's say that all this field is going really, really fast. That sometimes like I nap a while and then everything happens. So
Mm.
it's also complex to follow one trail. Probably I am excited for a lot of things for nothing at the same time, probably.
Right.
But definitely, maybe it could be once it's like the development of enzymes that can degrade plastic, not more for the this to have the ability to destroy it in nature because then it's too late, but to have options for increase the recycling of the plastic. So to have a biochemical recycling that can bring the plastic to the original bricks and to
Mhm.
make it from scratch to have it like other plastic that has characteristics closer to the virgin one.
Right. Um, how do you communicate complex biotechnology technological concepts to non-experts or policymakers? I think you're doing that right now, but
Yeah,
I guess it's
it's
important.
hard. It's two different targets definitely. So for non-expert, what I tried like to put in their clothes and
Mm.
to not give it for granted a lot of things and to have a way of communication that brings like excitement of the wow effect. Also, I like to be really from the theatrical point of view, and I did it at TEDx. And I try to be like shocking immediately at the beginning. It's in Italian that well as well, unfortunately. Um, with the policymaker, of course, it's different because they have a different language. Also, when we, as Bioeconomy Youth Ambassador, we wrote this Bioeconomy Youth Vision that is available online to try to engage with the Commission on how we would like the bioeconomy to evolve. And the difficulty that I saw, especially from the young one that are more on the activist side of the thing, it's to have a language that really talks to the policymaker. That I mean, you have to lure them inside the topic that you want to talk, and you have to talk about also politics. You have to talk about countries. You have to talk about economics. That
Mm.
from a person that is like strictly activist could look like stupid, let's say, because we have a crisis. So let's talk about that. Like for example, at the beginning, the a proposal of the initial part of the abstract was really like shocking. Like the reader would be really shamed of him or herself is not doing something for changing the things. And I suggested to change it because otherwise, if the trailer of a movie it scares you, except if it's a horror movie, of course, you don't go to watch the movie. So the goal of the abstract was to have the person, the attention of the person, and have this person read the document. If you scare immediately the person at the beginning, it goes away because he
Yeah.
has a short amount of time for you. Luckily, we happened to meet Maros Sefcovic, which is a High Commissioner of the European Commission and the Vice President for the EU Green Deal, and we delivered him this document. So we hope that he read it, and we keep it in consideration for the 2025 revision of the bioeconomy strategy. That definitely is now even more related to biotechnology because in the biotechnology initiative, biotechnology and biomanufacturing, that basically is fermentation initiative that was described by Ursula von der Leyen, there's inside the bioeconomy strategy change. So now even more biotechnology and bioeconomy are even more related. But often there's everyone is afraid to mention GMOs or genetic modification because we know that in Europe people are still afraid of that. So one of my goals as communicator is to try to fade off this afraid.
Yeah, but opinions can change, but politicians tend to follow the consumers. You see that with nuclear power, there's a bit of a change in the sentiment in some parts of Europe now. We are at the end of the time, so one quick question with a simple answer if you have. If you could fast track one, and this is a question that came in, one innovation in your field, fast track one single innovation, That's your dream breakthrough. What would it be?
The scale-up process for fermentation, from lab scale to huge scale, because
Right.
that
So
definitely.
simply, so pimp the machinery.
Exactly, exactly.
Okay. All right. Thank you very much. It's been very interesting for me. I knew something about the subject. I know a lot more.
Thank you, Lars.
Um, and I will still follow you, even though it's in Italian on Insta. Where else, shall people follow you on LinkedIn or Facebook also, or
Yeah,
where?
on LinkedIn mainly. LinkedIn and
LinkedIn.
Instagram are the biggest channels that I use.
Okay, I will. You will find me in the commentary from now
Good.
on.
Looking
All
forward
right.
to see you there.
Yeah. Take care.
Okay, bye.
Bye
Bye bye.
bye.